afternoon. We're going to go ahead and get started. Just a few announcements and reminders. So make sure to submit that integrity pledge and your commitments. Over half of you have already done it. Great, you're set. The rest of you, make sure you do it by the deadline after class next Thursday. Office hours have started this week. I think we had our first set last night, and my office hour will be this Thursday at 11 a.m. And then your first set of discussion sections will start this Friday on October 3rd. And to prepare you for that, there will be your first homework that will be posted after class on Thursday. Also, register your iClicker. If it's not registered, you're not going to be getting points for this class, so make sure you do that. A lot of you already have. In fact, well over 75% of you actually clicked and got that first extra credit point in class last Thursday. If you weren't here or you didn't, it's no big deal. Remember, there's a maximum of 10 points you can get, so there's many, many more classes in which you can earn that clicker point by participating in the polls. Does anybody have any questions about class format or things that I have here in the announcements? All right, so let's start off with a brief review of the molecular biology material we did talk about in class last Thursday. We said molecular biology really revolves around three basic macromolecules, the players, and then the processes of what they do or how more of them is made. And so what are the three players? One. Sure. More general than that. The three major molecules. Yes. DNA and protein, and what's in between? RNA, exactly. DNA, RNA, and protein are the three major molecules that are then made of constituents like nucleotides and phosphate backbones and so forth. And then there are three major processes, and so the process of DNA being copied into more DNA is called replication. And when our genes get copied into RNA, that process is called transcription. And finally, decoding of messenger RNAs into protein is translation, exactly. So those are the three major processes we will learn about, as well as all the regulatory steps that go into them. We also talked about in class how DNA is a genetic material. We went over a classic experiment that led to help establish this conclusion that in almost all cases, DNA is what is inherited and encodes for the progeny. We then did talk about the chemical composition and structure of DNA. So DNA is just going to be nucleotides held together by what kind of bonds? So the string of single-stranded DNA. A nucleotide is attached to another nucleotide by phosphodiester bonds, exactly. For DNA to be double-stranded, it's going to have anti-parallel, and what kind of bonds hold the bases together? Hydrogen bonds, exactly. And then finally, we talked about that often we need to denature DNA if we're going to copy it in the laboratory, or if your cells are going to copy DNA into more DNA, or to transcribe it into RNA. It needs to be denatured or melted or unwound. Those hydrogen bonds need to be broken. In the laboratory, we can just use high heat to break those hydrogen bonds. And we also said that there's this term, melting temperature. What does melting temperature mean? So it is a temperature at which how much of the double-stranded DNA is denatured? Half, exactly half. When half, that is your melting temperature. And we looked at some graphs of how we could measure that by taking absorption and then see how it's a function of GC and AT content. Any questions about what we talked about on Thursday? All right, let's get your clickers out. See how many people already have memorized their bases and nucleotides. So, of these options, which of these nucleotides are found in RNA? Yep, feel free to discuss, debate among your friends. Ten more seconds. All right, get your final clicks in. All right, we have a lot of people liking answer B, which is 3 of the different options of there, and we have some votes for a few of the others as well. All right, who wants to volunteer what their answer was and why they came to that conclusion? Yes. Yeah, thanks for sharing. Okay, let's talk through this. So if we're thinking of RNA, there's like 2 major parts of RNA we need to think about that kind of make it distinct from DNA. What is one thing you should look for? And it's something that was true for 1, 4, and 5. Yes?
Both of the OHs have, or both the 2 and 3 primes have OHs on them.
Right, so RNA is not deoxy, so you need to have those hydroxyl groups. So first thing you can do is look at your sugar, your base, and do I have hydroxyl groups at both 2 and 3 positions? That's true for 1, 2, no, that's kind of a weird one, right? 3, this would be your normal DNA deoxy because it is missing the hydroxyl group at the 2' position. All right, we have some hydroxyls at the 2 and 3 position here and also right here. So far, what we've identified, those 3, they have that component. But what is the other thing we need to think about that is a difference between RNA and DNA? Yes? Thymine versus uracil. So we need to be able to identify the bases and make sure that the base that is on that sugar that has both hydroxyls could be an RNA. What is this base in number 1? Looks like it can make 2. Yeah, adenine or adenosine, whichever you want to call it, it's fine. So yeah, adenine, that's an RNA. So yes, 1 is correct. We didn't choose 2, but does anybody recognize this base? It's uracil, exactly. So if this had the 2' and 3' hydroxyls, this would be found in RNA. It's not going to be found in a chain of RNA when it's missing that 3' hydroxyl. What is this base? Yeah, cytidine, right. So see, cytidine, that could be an RNA, but we're missing that 2' hydroxyl. So this is a DNA nucleotide. What about 4? We said 4, it has those 2 hydroxyls we want. What base is this with this methyl group? Thymine. So thymine has the methyl group. Would thymine be an RNA normally? No, thymine is in DNA. So 4, is that a correct answer? No. So once we recognize that this is a thymine base, 4 is not a correct answer. What about 5? What base is this? Guanine, right. You can see how you can make the 3 hydrogen bonds here. It has the hydroxyls. So this G, guanine, is found in RNA. So what is the final answer? D is the correct answer. So only 1 in 5 fit with what we need to recognize as being in RNA. Does everyone see that now? All right, just a few things I want to say about this then. some reason my slides seem to be stuck. And also, oh no, the Wi-Fi went out. Yes, yes. All right, so hold on here. Attempting to. Does anybody have UCSD protected back on? Yeah, I'm running a diagnostic, but this is not looking so promising.
Is there like one we have to plug in? Because we have the PDF pulled up. That's still loading.
Yeah, I'll be able to run the PowerPoint, but then the clickers aren't going to.
Okay.
It's not giving me.
It'll come back in a bit. It's okay.
Yeah, all right.
If anything,
So
there's a number here.
we are just going to. All right, so it seemed like the Wi-Fi was working when you all clicked in. So that should have all been captured and safely stored. So we don't have to worry about that until we come to the next clicker question. We can move forward, though, with the slides and continue class. So hopefully it will reconnect at some point. All right, so moving forward. Oh, why? It was. There we go. All right, so yes, so letter D, 1 and 5 were the correct answers. But I want to emphasize that the pre-midterm and the pre-final, as well as the midterm and final, are going to be multiple answer, multiple choice questions. So what that means is when you are given this type of question, it will give you all the options, and you need to make sure to choose all the correct options and none of the wrong options, right? Because the way Canvas grades is it will subtract for wrong answers. So only choose the answers that you are sure of as being correct and at least get partial credit, and don't lose all credit by just simply choosing all of the answers. Yes? You cannot get negative points. 0 will be the lowest you can get, and this is why it subtracts for wrong answers, because otherwise you would just choose all of them all the time, right? And then you would be assured to have some right. So what I have done on the Canvas site is under exams, I have put a practice online exam. I highly, highly encourage you to take it. It's only like 4 very simple questions, but it gives you the opportunity to practice choosing the right answer, multiple answers, making sure that the figure loads on your computer with the browser you're using. If you have not practiced this ahead of time, it will not be possible to help you during the time of the exam if your browser isn't able to use this. If you're able to see this practice Canvas quiz and run it, no problem, on your computer, then the pre-midterm and the pre-final will work fine for you. I see that some of you already took this this practice online exam. It's anonymous. I don't know who took it. It doesn't give you grades or anything, but I'm glad to see some of you are already taking this opportunity to make sure everything runs smoothly well ahead of exam time. If you do notice an issue right now, come talk to us. We will fix that before it actually comes to exam time. Any questions about that? All right, so let's move on to new material, if we can. There we go. All right, so tonight we're going to start off by talking about how we define a gene and then also what we mean by gene duplications. Because what we want to understand is, so this is called a karyotype. Karyotype is simply showing sort of the number, size, structure of all the chromosomes in an organism. What organism is this? Likely human, right? So We all have 22 pairs of chromosomes and either 2 X's or an X and a Y, or sometimes something even a little different than that. But somewhere in all of this, we have 6 billion base pairs of DNA. And so what is that DNA actually composed of? And so in our class today, starting in lecture 2 here, we're going to talk about these major types of DNA sequence. So what we consider to be genes, and then the parts of genes, and then also talk about these repetitive sequences that surprisingly make up a large majority of our genome. So to begin, we need to really make sure we're all on the same page with defining what is a gene. So what is a good definition for a gene? What is a gene? Sure.
Is it like a DNA sequence that defines a certain characteristic?
DNA sequence that defines a certain characteristic, or it encodes something, right? What is it? What do we want it to encode? Yes. A protein is a type of gene. Are there some genes that don't encode for proteins? Yeah, so Hox genes are protein coding genes, but they have regulatory sequences. Maybe that's what you were thinking of. And so our best working definition is actually that a gene is the entire nucleic acid sequence, the DNA sequence, that is necessary for synthesis of a functional product. A protein is one type of functional product, but there are also other types of genes that do not get decoded in a protein, but they most certainly make a functional product. So if we are talking about a gene, and this particular gene that we're looking at as an example would be a protein coding gene. It would have a coding region. Sometimes the coding region can be interrupted by sequences that need to be removed. What do we call those interrupting sequences? Introns. And what is the process that removes them? Splicing. And we'll have a whole class on understanding the mechanisms of splicing later in the quarter. In addition to the coding part of the gene, so the coding part of the mRNA is going to be what the ribosome translates into a protein. It encodes for a protein. But before that coding sequence, there is this what's called a 5' UTR. What does UTR stand for? Untranslated region, exactly. And what does that mean? It means exactly what it sounds like. This part of the messenger RNA is before the coding sequence. There's no start codon here. This is part of the mRNA. This is where transcription starts. And you'll get this sequence of RNA before you finally get to the start codon, where then the ribosome would start translating. The intron would already be spliced out, and it would get, when does the ribosome stop translating? When it reaches a stop codon, exactly. And we'll talk more about this when we have the class on translation. Well, once the ribosome stops and dislodges from the RNA, there's also sequence still attached to the RNA in this 3' region, the 3' untranslated region. Ribosome doesn't translate in there. That's what UTR stands for. So we'll have sequence at the 5' end and at the 3' end. And typically, these sequences can have regulatory effects on the messenger RNA, how stable it might be, how well it might be translated. Most or many protein coding genes also have a sequence at the very end that is called the polyadenylation site. What happens at the polyadenylation site? It's where the 3' UTR is going to end, and then what gets added onto it? A whole bunch of A's. So poly means many. So there's a string of A's. It's at the very end. So sometimes it's called the poly-A tail. And we'll also talk about that process in a later class. All right, so that would be the RNA, the initial RNA that gets made. But you need instructions for when and where and how much of that RNA to be made. And so those would be your transcriptional control regions. Transcription, remember, is decoding DNA to synthesize RNA. And so there will be what we call generally promoter sequences that will recruit the RNA polymerase to allow transcription and making of this messenger RNA to be synthesized. Any questions about these general parts of a gene and how you get a gene product? All right, so this then leads to a question that was alluded to previously. So we were talking about a protein coding gene, but what about sequences that encode for ribosomal RNAs or tRNAs? What are ribosomal RNAs? What process are they involved in? Translation, right? They are part of the ribosome. What about tRNAs? What does tRNA stand for? It's a good guess. Transfer RNA, because they transfer amino acids onto the ribosome for translation to occur. So both ribosomal RNAs and tRNAs are involved in the process of translation, which we'll talk about later in the class. But are these genes? Are the sequences that encode for ribosomal RNAs and tRNAs genes? Looking at our definition. Are these functional gene products? Absolutely. You would have no translation. You would have no protein synthesis if you didn't have ribosomal RNAs, if you didn't have tRNAs. So that's why our definition says a functional product. In this case, the functional product is an RNA that's not going to get translated into protein, but it is going to be part of the translation machinery. Is that a clear question? So if you're talking about like an oncogene or a tumor suppressor gene, those are usually proteins, and they are just proteins that are either regulated differently or have picked up a mutation that caused them to have a different function. They are a gene. They may be a mutated form of a gene producing a different kind of gene product, but absolutely those would be genes. Question? So transcription factors are what kind of molecule typically, or always? Proteins, right? So protein is a functional gene product, right? So absolutely the genes that encode a transcription factor are genes. Other questions? All right. So here's a big table from your book that lists all the major classes of eukaryotic DNA and how they're represented in our human genome. And so protein coding genes, what we most often think about, so encoding transcription factors or ribosomal proteins or collagen, these are all protein coding genes. And they can range in size. Some encode very small proteins, some are huge genes. And in the human genome, we have an estimate about 20,000 protein coding genes. And they take up a good portion of the human genome, the actual gene, which would include the introns, it would include the UTRs, the promoter sequences that allow the messenger RNA for that protein coding gene to be synthesized. And so let's talk about some general types of protein coding genes. So a solitary gene is a gene that's just found once in the genome. It is a protein that is distinct. It doesn't look highly homologous to anything else in the genome. And if it has a very important function, if you get a mutation or you lose that gene, it's probably not going to be viable. But many genes in our genome actually have arisen through duplication events. And we'll look at how that can happen in a moment. And what duplicated genes are, they are multiple genes of close but not identical sequence. They're often homologous or related or similar to each other. Many times they're close to each other, and so could be on the same portion of a chromosome. So this example here on chromosome 11 has these duplications of the human beta-globin gene. And we call these duplicated genes part of a gene family because they are related to each other. So let's look at this example here of the human beta-globin gene. What is beta-globin? It's a protein. What is it involved in? Beta-globin. Sure. in all of us, right? We all need hemoglobin in our red blood cells to transport oxygen, right? And hemoglobin is composed of two beta proteins and two alpha proteins. So these beta-globin genes are essential for your body to have red blood cells that can do their job and transport oxygen. And what's really cool here is we've had, over the history of human evolution, duplication events happen that we have multiple beta-globin genes here that have taken on slightly different properties depending on what time they are expressed and what they need to do. So right now, you and I, we're adults, so we are expressing HBB and HBD. So these are adult hemoglobin, beta-globin genes that are really good at depositing oxygen in our tissues. When we were embryos and at the fetal stage, we actually expressed different forms of the beta-globin gene, these duplicated versions that have taken up slightly different sequences. They encode proteins with slightly different amino acid sequences, and they actually have slightly different oxygen-carrying properties. These, the fetal and the embryonic, are actually much better at stripping the oxygen from the maternal red blood cells so that it can be deposited into the rapidly growing fetus. And so by these duplication events, we have different members of this beta-globin gene cluster that now are expressed at different times in development and have different oxygen-carrying properties. All right, so how do we get these duplications? And so as you know, during meiosis, when you're making gametes, there's crossing over of chromosomes that happens, right? And the crossing over requires requires homologous sequences. So you could have two chromosomes line up equally and have an equal crossover event. But if there are duplicated sequences or these repeat sequences that are the same, so this pink box here, this L1, is exactly the same sequence as this pink box here, this other L1 is our type of repeat sequence we'll talk about later in class. And so instead of this lining up here and this lining up here with equal crossover, you can then instead get these two line up, because they're the same sequence, and get what we call then an unequal crossover event. And the result of that during recombination will be that this portion will cross over and pick up this portion of the chromosome. And now what do we have? Now we have two copies of this globin gene on one chromosome, and we have the other chromosome with no copies of it. This is going to die out of the population. The globin gene is essential. But now you have a chromosome that has two copies, and so now you have room for one to maybe change a little bit, and if that change is beneficial, it will be maintained within the population, and that is what's happened with our globin genes. And so the results of gene duplications, as in the case of our beta-globin gene cluster, is you can have beneficial mutations. So when we hear the word mutation, we always think bad. Mutation just means a sequence change. And in this case, these sequence changes have been beneficial. It allows, the embryos to extract oxygen from the mother better. Probably the embryos grow faster, more healthy. That is going to have an advantage. The adult hemoglobin's great at depositing oxygen throughout all the tissues in our big bodies. That is beneficial. But the other thing that can happen, because these sequence changes are random, right? So it's just a random change. And if that change is beneficial, then it's maintained in the population, like these hemoglobin genes here, or these beta-globin genes here. But many times that sequence change will lead to a non-functional gene product. It could mutate the start codon, or change a splice site, or do something so that you no longer can encode a functional gene product. And so that is what this is depicting here. We call those pseudogenes. Pseudo means sort of, not exactly, so it's not exactly a gene. And so this beta 1 here, this no longer encodes any kind of beta-globin gene in this cluster. If the pseudogene doesn't cause any harm, there's no pressure to get rid of that sequence. It's just sort of like stays there, and we see these throughout our genome. These pseudogenes that no longer produce a gene product like the genes that they were duplicated from. However, I should mention that we are learning that sometimes pseudogenes RNA still gets transcribed from them, right? But let's say there's no start codon. It's not going to be decoded into a protein. But you're still making RNA, and can RNA sometimes do things? Yeah, and so some of these pseudogenes, people are starting to think, especially ones that are highly expressed, the RNA itself could have a function that could be good or bad depending on what that sequence actually does. So there's a debate about what we really mean by pseudogene, because some of these might actually be doing something. Question?
I didn't capture what do you mean by beneficial mutation.
Yeah, so what we said here is there are slight amino acid differences, so there's slight changes in the DNA, so mutation DNA has changed. That DNA then encodes for a different sequence RNA that encodes for a different sequence of amino acids. In the case of the fetal versus adult, the part that holds on to the oxygen, those amino acids hold on to it tighter in fetal, and then it's different amino acids that can let it more loose and deliver to the tissues. So that's a benefit for the human species, right? It's a beneficial change that allows these different globins to take on different properties that help the organism thrive. Does that make sense?
By mutation, you mean like you don't know how it's happening, but it's happening? Like
So mutation, so it's a random sequence change, right? So every time your DNA gets copied, there's a chance that there will be a change in the sequence. And we'll talk about DNA repair next week, but if that repair doesn't happen, that change stays in the sequence. That's how mutations happen, right? Yes? Yes, sure. It is one of the reasons why we have gene duplications, yes. Sure. It would definitely be a change from the ancestral sequence. So yes, that would be a mutation. Anything else? All right, so those are examples of duplicated genes. And when we're talking about duplicated genes, usually they have slightly different sequences and slightly different functions. And that is to contrast into the next type of repeated genes that we're going to talk about. And these are examples of tandemly repeated genes. What does tandem mean? Yeah, one after the other. And repeated means the sequence is repeated one after the other. And the difference here is that tandemly repeated genes encode identical or nearly identical gene products. And some examples are ribosomal RNAs, tRNAs, histone coding, those are protein coding genes. What's one thing that's in common with these types of tandemly repeated genes? We have some RNA genes, we have histone genes. Your cell just need a few of those. No, you need tons of these, right? We'll talk about on Thursday how histones are the core constituents of your chromatin. All your DNA needs to wrap around the histones. You need tons of histone proteins. For translation to occur, you need tons of ribosomes, tons of ribosomal RNAs, as well as the transfer RNAs that participate. So one way that your cell has solved the problem of getting so much of these gene products is just by duplicating the genes that need to encode these. And so I'll show you an example of where this can get you with the ribosomal RNA gene. So suppose we have just one copy of the pre-ribosomal RNA gene encodes 3 different ribosomal RNAs that it will get spliced into that you need for any ribosome to function. And so you have that gene and you have one polymerase. Sit down and start transcribing that gene. Well, based on how long that gene is and the rate of transcription, you could produce about 288 molecules in 24 hours. Okay, that's a lot, But if you're a rapidly dividing cell, dividing every 24 hours, like a growing embryo, you actually need more like 5 to 10 million ribosomes and therefore ribosomal RNAs in order to carry out those rapid cell divisions. All right, so that's certainly not going to be enough. So how can we get more? Well, it turns out that if you have one gene, that multiple RNA polymerases can start transcribing the same gene one after the other. So what you're looking at here is in the middle here is the DNA that is being transcribed. And an RNA polymerase molecule is starting to make RNA. And so at this end, this is the beginning. So the RNA molecules are very short, and then they're longer as it gets closer to the end of the gene. So basically, as soon as the first RNA polymerase clears the transcriptional start site, the next one comes on and starts transcribing. And so if you can have multiple polymerases transcribing at the same time, based on the length and the size of the polymerase, you can have about 250 on this gene. It's going to get you up to 70,000 in 24 hours, still not nearly enough. So the way the cell solves this problem is you actually have more like 100 pre-ribosomal genes, and many of them are in tandem. So this is a ribosomal RNA gene, this is a ribosomal RNA gene, this is another one, and they all have polymerases starting to transcribe them one after another. And so if you do the math, this then gets you up to about 7 million ribosomal RNAs that you'll need within that 24-hour period. Any questions? All right, so now let's move on to for what we recognize as the functional gene product coding parts of our genome, to what has been more classified as the sort of non-coding or non-functional part of our genome, because it's actually a very large fraction of our genome. And so we'll talk about this abundant, what has been considered to be non-functional DNA, and then the simple sequence DNA repeats that we see in this sort of non-functional repetitive part of our genome. So if we look at our entire genome and we map out the sequences of what we know that they are composed of, what we see is, so the exons are what have the coding information, right, to make a protein. This is a very, very small fraction of your entire genome. It's actually only about 1%. In humans, our introns are actually pretty big, so they take up a pretty big fraction of the messenger RNA that is being transcribed. And then we're going to have promoter sequences, right? So they are not transcribed in the typical sense, and so that would be included in your other intergenic DNA. And then there's a whole bunch of repetitive DNA sequence throughout your genome as well. And so really, the part that we study and think about the most is a very, very small fraction of our genome. All right, so 99% of our genome is non-coding, but how much of the genome is actually transcribed? So we know, are introns transcribed?
No.
What happens to introns? They get spliced out of what? An RNA. So they most definitely are transcribed. Transcription is producing the initial RNA molecule. And in that RNA molecule, you're going to have 5' UTRs and 3' UTRs. And then you're going to have those introns that get removed, and then everything else, including the UTRs, is part of your mature messenger RNA. So, all right, so we know that so at least 25% is transcribed. What about the rest? Is there transcription events happening in other parts, in the repetitive parts? And so we used to think not so much, but now in the past decade or so that we have very sophisticated sequencing technology, we see that almost the entire genome produces some level of RNA at some tissue or at some time. And so there's still debates of how much the actual, is it 80%, is it 95%, is it 100%? I think that's not quite as important as this question is, all right, so we're having all this additional RNA product coming from our genome, but how much of it is actually functional? And that is still a question that is debated. You know, we know that our protein coding genes can encode for a functional gene product. We know that genes that encode ribosomal RNAs, other types of non-coding RNAs, those produce functional gene products. But there's other repeat regions, for example, and transposons in our genome that sometimes also get transcribed. And so do they make functional gene products? Do they do anything to affect our physiology or health? And we'll touch on that a little bit at the end of this lecture. So the answer to this question is, I don't think we know yet. All right, so we're going to talk about now the repetitive DNA. And so repetitive DNA is going to include very simple sequence repeats, as well as when we get down and talk to transposons. And some transposons themselves actually have the capacity to encode for proteins to allow for their own mobilization. So they do make functional gene products. And so this is what layers the complicated nature of figuring out what genes we have in our genome. All right, so when we're talking about simple sequence DNA repeats, there's two basic types that your book talks about. So there's what are the longer repeats. These are called sometimes satellite DNA. They can be repeats that are quite long. So you might have 500 nucleotides of unique sequence, and then that 500 is repeated, and that 500 is repeated. In some cases, it's just 14 to 20 nucleotides, but then it's repeated in tandem. We said tandem means one after the other. For large chunks, so 20,000 to 100,000 base pairs long of these repeated satellite DNA sequences. And these regions are primarily found at two particular places in our chromosomes. One is at the centromeres. What do centromeres do? What function do they play for chromosomes? Centromeres. What do your chromosomes need to do after they're replicated? Yeah, so equal division, especially if your cell is dividing, you want them equally divided into the daughter cells. And then telomeres. So telomeres are found at the ends of your linear chromosomes. What's the general job of a telomere? Protection. And we will talk about how telomeres are synthesized as well as how they actually do this protective job in a class next week. All right, so then much smaller simple sequence DNA repeats are called microsatellites, micro, small. And these can be just one nucleotide, so AAA, or up to 4 base pairs repeated over and over again. Again, they'll be one after the other, usually up to 150 repeats, although that can certainly vary. And these are important because sometimes you may have heard of these repeat expansion diseases. We'll look at some examples in a moment. And so this is an example of a 3-nucleotide repeat. So there is CAG is the number of repeats, and it is repeated 1, 2, 3, 4, 5, 6. So in this case, there are 6 repeats of CAG. And so some examples of these repeat expansion diseases, some of them I'm sure that you have heard of before. Probably one of the most famous is Huntington's disease. So this happens, so we all have a Huntington's gene. We all have the HD gene in our genome, and we all have some repeats in this gene in our genome. CAG repeated hopefully only 6 to 35 times. If you're within that window, that doesn't seem to be associated with disease at all. But once you go a little bit higher in the repeat number, this is then what's associated with Huntington's disease. Once you have this repeat more than 36 or more times, so CAG encodes an amino acid, glutamine, and if you have too many glutamines in a row, that causes the protein to fold differently, get sticky, stick to each other, clog your neurons, and leads to death. Here's some other examples of some diseases that have, interesting, they are all are CAG repeats. They are all in coding regions, so the part that gets translated. So you get multiple in a row of the amino acid encoded by CAG, and they are in various different genes and cause different diseases. You can also have expanded repeats in non-coding parts of a gene. So in this Fragile X syndrome example here, the repeat is actually in the 5' untranslated region, and that changes the expression of the gene, and that then leads to the disease situation here. You can also have expansions in even introns that can change splicing of a gene and therefore lead to the disease state. So here's just an illustrated example of what we mean by this repeat in Huntington's disease. So we all have this HD gene in our genome. There's a coding sequence. I took out the introns. They're not illustrated here. But towards the end terminus or beginning of the coding sequence of the Huntington disease gene, this is where we all have this CAG repeat, 6 to 35 times, repeated over and over again. This encodes, what is Q? What amino acid? Glutamine, yes, glutamine. So each CAG encodes for a glutamine, and so you may have 6 or you may have more. But in patients with the disease, you then have an expansion of this CAG repeat up to over 100 times. And the severity of the disease is associated with how many repeats you have. That will be associated with the likelihood of this misfolding and causing the aggregates. And it's a devastating disease, I'm sure you know. And so here's an example of what a cross-section through a normal brain looks like versus a poor person who is suffering from this disease where their neurons are all dying because of these aggregates of Huntington with the repeat expansion. All right, let's look at another repeat expansion gene with, let's try to do a clicker. Has anybody gotten back online yet? Yeah. Yes, okay, let's. Oh, no, wait, that's not what I want to do. Let's see if this is working now. All right, seems to be working. All right, so this question is talking about myotonic dystrophy. So your muscles deteriorate, really horrible disease. And this is caused by a CTG expansion in the 3' UTR region of the DMPK gene. And I'm telling you, this is associated with altered splicing of many cellular pre-messenger RNAs, but not DMPK pre-mRNA itself. So here's the mRNA once it's been transcribed. So you have U instead of T because the RNA here. And you have tons and tons of repeats of this CUG in the 3' UTR of this messenger RNA. So based on this information, which of the following describes a possible hypothesis for how 3' UTR expansions in DMPK may affect splicing of other pre-mRNAs? And it's recording all your clicking, so I think we're in good shape now, hopefully. 10 more seconds. All right, put in your final answers. Oh, all right. We like all the answers, right? All right, let's go through this and think about what is going on here. What could explain this? All right, who wants to offer an answer, an explanation? Sure. Oh, you can say whatever you want.
I chose C because, from what I understand, it's in the UTRs, so it's not really coding for any proteins. And then also, it would make sense that like the RNA building thingies are like they're like binding to the excess of the CUG chemically. That makes sense.
All right. So yeah, so you said a few key things there. So first, I think you narrowed it down. You didn't like the answer A. And so why don't we like the answer A? Well, where are the repeats? They are in the 3' untranslated region. Can that change protein sequence? No, it's not translated. It's after translation has stopped. So you are not changing the protein sequence. But what about B? The DMPK protein may play a role in splicing. So what's encoded by this mRNA may be a splicing factor. And the repeats change the protein levels. Can UTR sequences be regulatory and affect how stable the RNA is for translation or how well it's translated? Yeah, so the UTR sequences can be regulatory. Those sequences can be recognized by RNA binding proteins that can either stabilize that messenger. If you have a lot of the messenger RNA, then you can make a lot of protein. If you block the ribosome from seeing that messenger RNA, you're going to make less protein. So you'll see examples later in class of how the UTRs can regulate the level of the RNA or how well it's translated. So we have changes in a UTR. Could that affect the level of DMPK protein that you end up with? Sequence will stay the same, but maybe you'll get a lot more or maybe you'll get a lot less. So B is actually an option. But then we'd like C too. So let's see, does C make sense? So our friend told us that proteins involved in splicing may bind to these repeat sequences and be sequestered away from their normal target mRNAs. So this one is a little bit tricky to think about, but you can imagine if you're going from just having a few repeats that are recognized by an RNA binding protein, a protein that's needed for splicing, and then you suddenly have way more of those repeats, all of those proteins are going to get soaked away and stuck on this abnormal RNA instead of doing their job of splicing. And that actually is the what we now know through experiments, what actually is happening in the case of these patients with these repeats. But with the data I gave you, B or C were compatible, and therefore E was the ultimate correct answer for that. And if you don't get that sequestering away idea, I do have an illustration here. Oh boy. There we go. Okay. So you can imagine in the non-disease state, you have the 3' UTR of DMPK. It turns out these CUG repeats kind of fold back on themselves and form sort of a partially base paired structure. And there's a protein called MBNL1 that likes to bind to this sequence and structure. And so if you only have a few repeats, then maybe just one of these proteins is going to be attracted here. And so you have a lot more of the protein available to carry out splicing of all the other messenger RNAs that need that factor for splicing. But now, if you go and repeat this sequence and have a lot more binding sites for that specific protein, it's going to sequester or titrate or compete for binding to that MNBL1 protein. And now there's very little left to go around and do all the splicing that it needs to do. Does that make it clear? Question?
So are UTRs spliced as well as introns after the mRNA is transcribed?
So that's a good question. If a UTR has an intron in it that's in the way, the intron gets removed, but the UTR sequence remains part of that mature messenger RNA. So here's a question for you. So would they be considered exons then? So when we're talking about splicing, there is only introns and there is only exons. The introns are in the way and they are removed. Everything else that remains is ligated back together and is considered exon. So are UTRs exons? Yes, absolutely. Other questions? All right, and if you're interested, there's this very short YouTube video that actually talks about this process and this disease that I would recommend. And I should mention, so all of these clicker questions, as well as the additional things I show you, I'm going to post them after class on Thursday for our first 3 classes. So you'll have access to all of that. All right, so now let's move into the last type of repeat sequence we're going to talk about in class, and these are transposable elements, also called mobile DNA, because it was initially described as moving DNA elements. So I have pictured here a very famous person who actually won a Nobel Prize in 1983, and she came up with the idea of mobile DNA by studying, what are these pictures of? Anyone know? Corn kernels. And who is this? She discovered and first described transposons. Yes? Yes, Barbara McClintock. Does that ring a bell for some of you? Yeah, so she, way back in 1953, first proposed this idea that there were DNA elements that were hopping around her corn kernels. Everyone hated this idea, right? Because it's 1953, we finally figured out that DNA is the genetic material. It's this stable stuff that allows for transmission. It can't be just moving around. And so nobody believed her for a really long time until finally they came around and realized, oh yeah, and in fact our own genomes are composed of a lot of these transposons. So it took 30 years, but she finally got the Nobel Prize for this amazing discovery. And she discovered it by studying corn kernels and phenotypes in corn kernels. And so the corn she was studying, the kernel, the wild type kernel is purple. There's a pigment gene that encodes for a purple color in this corn. And then every now and then all of a sudden she'd get a totally white kernel. It's like, oh, something has happened to that pigment gene. But the weirder thing is, she could also then get kernels where some of the cells reverted back to the purple color. They got their purple pigment back where other cells were still white in this. And by looking at chromosomes, she could see evidence for there seemed to be some parts of the DNA moving around. And what was happening was a transposon was jumping into the pigment gene, disrupting it. You can't encode the pigment gene, and so therefore the kernel is white. But every now and then you could get that same element to jump out of the pigment gene. It comes back together, it is mended, and now it can encode purple again. And so this is one of the first examples of a DNA transposon. And so there's 2 types of transposons in general. We'll talk about DNA transposons and compare how they are different from RNA transposons. And another word for RNA transposon is a retrotransposon. Retro because they go through an RNA intermediate. And then we'll talk about 2 major types of RNA transposons in our own genome, and these are lines and signs. And then think a little bit about the roles for transposons and these mobile DNA elements have had in evolution. And more recently, we've been thinking about perhaps some transposon mobility contributing to aging as well as diseases. So there's lots of examples of transposons that have been studied. Some of you might be familiar with the story of the peppered moth. Does anyone know what happened here? So back before the Industrial Revolution in England, there were all these beautiful little peppered moths that actually camouflaged really well with the types of trees that were available. Industrial Revolution happens, and what happens to the environment? Soot everywhere, right? So trees get covered with soot. These guys, these white mottled ones, stand out. But some lucky moth was fortunate that a transposon jumped into one of its genes that was responsible for this coloration and now turned it all black. Those moths then took over, right? Because they could camouflage much better in these soot-colored trees than the original ones. And this was because of a mobile DNA element jumping into one of these genes. Question? Yep. So jumping genes, mobile, so jumping, mobile, transposons. As you'll see, jumping maybe isn't the best word for the RNA types of transposons because they go through more of a copying step, but we'll see why that is. So we'll see why I don't like that term in a moment. Question?
So is this occurring during replication and gamete production, or is it like in a live present cell?
So it would have had to have happened in a gamete, right? So to pass, if it just happens in one of your skin cells, it's not going to get passed on into anything. So it has to be in a germ cell that can then get passed on to the progeny. And that progeny that got it was camouflaged really well. Its progeny are going to be all camouflaged really well. And so very quickly, this can get taken over because of the environmental change. So survival of the fittest is what happened right here. Is there another question? Yeah, wine. Many of us probably would like wine, right? Well, so the reason why we have Chardonnay grapes is they actually came from Cabernet grapes because of a transposon event that jumped into a particular gene. And then, of course, over people propagating this and other varieties, we now have a very nice alternative type of wine. In the past decade or so, we've actually recognized that transposons actually are responsible for a lot of different changes in the way genes are expressed. So not necessarily jumping into a gene and changing a pigment, but jumping into regulatory regions like UTRs or transcriptional control regions like promoters that will just slightly shift how much of a protein coding gene gets expressed or when in development it gets expressed. And when we look at our genome compared to a chimpanzee, we know we're like almost 99% identical, especially if you look at the protein coding genes. And we think that especially what defines differences in our facial structure has a lot to do with transposons jumping into regulatory regions and changing when and how we express the bone structure, the muscle structure, and all of this that causes these facial differences. There's also a really interesting example in your book. Some of you may have seen this, of this, one of the problems we're having with antibiotic resistance. So sometimes these resistance genes have moved into, or transposons have picked up resistance genes, and then they're able to jump into the genome of other bacterial species. And so they're transporting this resistance phenotype. And this is one of the big problems in antibiotic resistance. And so transposons can be great. They can give us Chardonnay grapes, but they can also be very problematic. And their whole goal is just to propagate themselves. They don't really care about us, right? So let's talk about these two different types of transposons and how they mobilize around our genome. So we have DNA transposons versus RNA transposons, which are also called retrotransposons. And the big difference is that DNA transposons always move by a cut and paste method, whereas RNA transposons use a copy and paste mechanism. So what does this mean? Well, just as the term suggests, cut and paste is if you have a DNA transposon on a particular chromosome, it will actually be cut out of that position of the chromosome, mobilized, and jump into a different position. So the former transposon site no longer has that transposon in it. That's how Barbara McClintock's corn went back to being purple, because it jumped out of that pigment gene. Pigment gene could then be expressed again. So you are cutting a transposon, a DNA transposon will be cut out of its original site and then move in to its new site. So that fits very well with jumping genes. It is jumping from one place to another. RNA transposons are a little bit different. They stay in their original location. They get transcribed by the cellular RNA polymerase into an RNA intermediate. So the original donor transposon, this retrotransposon, it is DNA. So all transposons are DNA. It's just how they mobilize. And the way RNA transposons mobilize is you need to transcribe into an RNA. That RNA needs to get reverse transcribed. What do we mean by reverse transcription? It's copying what into what? It's copying RNA into DNA, exactly. Because normal transcription, if you're transcribing your gene, you're copying the DNA into RNA. Reverse transcription is this RNA intermediate gets copied into DNA. It's made into double-stranded DNA. And then that new sequence of DNA will then insert into a target site somewhere else in the genome. So what's happened here is this transposon, this RNA transposon, hasn't jumped. It hasn't really mobilized. It's made a copy of itself. It stays in its original position, but then that copy gets reverse transcribed into DNA and moves into another position. DNA transposons, cut them out, paste them somewhere else. All right, so let's look a little bit more into the mechanism used by DNA transposons to mobilize. And so sometimes DNA transposons are also called insertion sequence. It's the same thing. So everything in pink and red here is the DNA transposon. It is the whole region that is going to get cut out and move somewhere else. For a functional DNA transposon, they actually will encode the protein needed to cut themselves out and to mobilize. And the general name for this is transposase. And so what you will find flanking on either side, so these black boxes here, these are not part of the transposon. They are the relic that shows a target site direct repeat that indicates that this sequence in between is the transposon that moved into that new site. These target site direct repeats are the hallmarks of just about every mobile element. And you'll see why that is with the mechanism used by transposon for mobilization. So when I say a target site direct repeat, if the sequence here, let's say, is 5' CAG, what's the sequence going to be here? What would a direct repeat be of 5' CAG? 5' CAG. That's what we mean by a direct repeat. It's directly the repeat. All right, so for a transposon to mobilize, to be cut out and pasted somewhere else, you need this transposase activity. And there's 3 functions of this type of enzyme. The first thing it needs to do is cut the transposon out of the original site. And it does this using a blunt cut, which means it goes directly through the double-stranded DNA. There's no overhangs. And so now you've freed up the transposon. But that transposon needs to be inserted somewhere else at a different location. And so to open up the DNA at another location, the transposase actually makes a staggered cut in the target DNA. What do we mean by staggered? We mean that it will cut here and here. And so you will have, therefore, some overhang, some single-stranded overhang. That's what we mean by a staggered cut. The last thing that needs to happen is a transposon needs to be ligated or inserted into that opening there. All right, so we have our transposon that has been ligated onto either end of this single-stranded region of DNA, and then we just need to fill in the gaps here. We need to remake the double-stranded DNA, and this is where we get these target site direct repeat sequences. So let's think about this. Let's say this sequence right here, so it just got pulled, it was base paired here. This sequence right here, let's say it's AAAA. If this is a bunch of A's, what is the sequence down here? Bunch of T's, right? All right, so if this is a bunch of T's down here, what is the sequence that's going to get filled in? Bunch of A's, right? We said these are a bunch of A's. What's going to get filled in down here? T's. That's where you get the direct repeat. So that's a simple example. Try putting in like CTG or something more complicated. You'll see it always works. That's how you get the target site direct repeats. And so that is how a DNA transposon will mobilize. Now let's talk about RNA transposons. And so the first one that we're going to talk about here are those that are called LTR retrotransposons. LTR just stands for long terminal repeat. And so in light green here, it is part of the transposon. There's just a long terminal repeat. So terminal, it's on the ends, it's long, it's repeat sequences. And then for the retrotransposon to be able to mobilize, remember, these transposons, these RNA transposons are part of the DNA. They are not going to get cut out and move. Instead, their RNA is going to get reverse transcribed into DNA. The DNA becomes double-stranded, and that new double-stranded DNA is going to insert somewhere else. So the activities that are going to be needed for an RNA transposon to mobilize, to put a new copy somewhere else, is you will need it to encode a reverse transcriptase, the enzyme that copies RNA into DNA. And then you need an enzyme like an integrase, which will cut open the recipient site for the DNA, open it up, and then allow for ligation of the reverse transcribed double-stranded DNA. Right, and so this is just defining what those activities do. As we said before, reverse transcriptase, sometimes abbreviated as RT, it's going to copy the RNA into DNA, and then integrase will insert the DNA into the genome. Look what we have here. We have target site direct repeats on either side. So what kind of cut must integrase make for where this DNA is going to be inserted? Staggered, exactly. All right, so those are a type of RNA transposon. There is another type of RNA transposon that's simply called those that are non-LTR retrotransposases, meaning they don't have those same types of long terminal repeats. But they're very similar. They also need to go through an RNA intermediate that gets reverse transcribed into DNA, and then the DNA integrated into a position. Our own genome has a lot of these types of non-LTR retrotransposons, and they are generally called either lines or signs. And that's basically how long or short they are. So line stands for long interspersed elements. Interspersed means they are scattered throughout our genome. The longest ones are the ones that are able to provide mobility, actually will encode proteins that have that reverse transcriptase activity and allow for integration. So ORF1 and 2, ORF that stands for open reading frame. So lines, the full lines will encode proteins that will allow for mobility. And so the longest ones are quite long, about 6,000 base pairs long, and they're a huge fraction of our human genome. The vast majority of them are not full. They aren't the full length. They don't still have the protein coding ability. But there's about 100 or so in our genome that do still have this protein coding ability. And so we'll come to that in a moment. And then in contrast to lines, we have signs. Sign stands for short interspersed elements. Sometimes these are also called ALU elements because of a sequence they have in them. The signs are so short that they don't encode their own proteins. The only way a sign sequence can be copied, can be reverse transcribed, and then integrated somewhere else is if there's an active line. An active line protein can take these sign sequences, copy them, and integrate them elsewhere. And they also have duplicated or have scattered throughout our genome and are a large part of our genome as well. And so I think the last thing that we will end with, and we'll go through the rest of the remaining notes on Thursday, is just to review and make sure you understand that there's two general types of transposons we're talking about. So there's the DNA that is going to go through the cut and paste, meaning it leaves its original site, goes into a new site. You're going to need integrase to do that. Do you need reverse transcriptase for this to happen? For DNA to jump out and move somewhere else? Do you need normal RNA transcription to happen? Yes, to make what? The protein encoded, to make that transposase, that integrase, to allow that to happen. RNA transposons are going to go through a copy and paste mechanism. So the original sequence will stay there, and then the new copy will be inserted somewhere else. All right, that's it for today. We'll pick up on the rest of this on Thursday. Hopefully the internet will work.